So Stephen has just been showing us distributions, and I will discuss how variation can affect the distributions. So biological variation is the phenotypic difference between individual organisms or groups of organisms due to genetic differences, environmental influences, or variation due to the interaction between environment and genotype. Phenotypic differences could be height, weight, body shape, and so on. In this picture, you can see, although the same species, there are clear differences in size and hair types. Technical variation occurs due to measurements taken, instrumentation used, and sample preparation, so variability results from systematic and/or random errors, which may be reduced by standardization of experiments. Badrila carried out a mathematical experiment to observe changes in p-value as technical variance increases. They used 100 independent simulations for the different technical variance levels, and assumed preset absolute values for two groups, the control and treatment, where the biological variation had a standard deviation of 15%, and as I said, a technical variance whose level varied across simulation. T-tests were completed on each simulation and p-value calculated. The figure shows a section of the results. The threshold p-value of 0.05 is indicated by the red dotted line, where values below this are considered significant. In every case, when there is no technical variance at x equals 0, we always see a technical variance below 0.05. We can see that the red dot, the average simulation, is below the threshold and p is statistically significant between the two groups. The p-value distribution is not seen. However, we very often have technical variance in addition to biological variance. As technical variance increases, p-values increase, and their distribution increases with average simulation approaching the threshold. At 0.2 technical variance, the average simulation is above the threshold. This figure shows how a small change in technical parameter changes p from statistically significant to non-significant, and highlights how sensitive p is to small changes in variance. With no change in sample size or underlying parameters, as technical variance increases, and added to biological variance, we get a range of p-values in the simulation experiments, so placing less trust in the robustness of p-values. Added to this is the use of incorrect biological variation data. A 2015 study looked at the reliability of biological variation data on the Westcard website, an online database, and highlighted many factors decreasing data reliability. The review showed that much biological variation data is outdated and comes from only a few, and sometimes just one paper. So we should think about how this incorrect data, and maybe not even acknowledging variation, can affect p-value. Increased variation can increase p-value distribution, and when considering that levels of both technical and biological variation will be specific and variable to a given setting, the impact on p-value distribution is clear.
So, although it would be impossible to completely remove the variances that Katrina's just mentioned, it is important to account for their impact on the p-value when designing a study. So if you could please pause the video and fill out questions 8 and 9 now. Okay, so can we use power to limit these vulnerabilities caused by the variances? Well, to utilise power during study design, the sample size, effect size and levels of variances should be understood. Power is the measurement of an experiment's capacity to provide a statistically significant effect and can determine the probability of the experiment rejecting the null hypothesis. The usual size of power is set at 80%, meaning that 80% of the time the null hypothesis is correctly rejected. A fundamental factor within study design is the quantification of the sample size of a study, also known as the N number. This is important for divulging not just the p-value and effect size, but also the power of the study. To determine the number of subjects required in the study, it's important to know the estimated effect size, variability of the expected results, and the potential shape of the data, be it binary or logarithmic. As seen within the figure, a much larger sample size would be required to reach lower p-values of less than 0.05, although the theoretical sample sizes shown in this figure could be seen as unrealistic. Yet, a larger sample size will allow the researchers to account for biological and technical variation within the experiment, and therefore improve the power. However, the idea of what constitutes a large sample size may vary, with a sample size of n equals 10 sometimes thought of as large. This indicates an underlying issue within study design and the difficulty planning an experiment with a large enough sample size to protect against sampling error stemming from the variances. If in fact the misunderstanding or misuse of statistics are the key element leading to the reproducibility crisis they seem to be, then the greater priority could be placed on their implementation. However, with the noted dichotomization of p-values at the 0.05 threshold and a potential overemphasis placed on this value, it seems within the results of some studies there may be an unreliable accounting for the variances, or even none at all. If pre-study calculations are the important factor they seem to be, then it could be said that improving the power of an experiment through increasing the sample size would be an answer to this conundrum, therefore leading research towards a brighter and more reproducible horizon. Okay, and please could you fill out the final question in the questionnaire, and thank you very much for listening.